gather your workstation with your buffers, your protein samples, all the tubes you're going to put your purified protein in, samples, pipette, and a protocol. So now you're going to resuspend your cell pellet using the lysis buffer. It should resuspend quickly and efficiently. Make sure you get all of it and transfer it to a tube. You can continue to do this for all the samples. You can see that that sample is likely GFP based on how green it is. So now that we have all of those set up, I'm going to add a little bit more of the lysis buffer or the wash buffer. You can add them in either order. Once I do that, I have my tubes, make sure they're all set up. And I'm going to set a timer for ½ an hour, and I'm going to set them on a nutator. You can use this type or a flat nutator. The 30 minutes is done. So now I have my lysed cells. I'm going to take my tubes and I'm going to spin them in a centrifuge, a balanced centrifuge, for 30 minutes. You could do this at 4 degrees or at room temperature. So while they spin, you want to make sure that your columns are prepared. So now we have columns prepared here in this rack, set up on a ring stand over a beaker. So here I am adding a little bit of wash buffer to just make sure that the filters are working. Then I'm adding in the nickel and TA beads. My proteins are purifying, or they finished spinning. So now when you get these out, you can see the GFP is clearly in the supernatant. When you look at your protein samples, they should look somewhat clear. So now I'm going to add 500 microliters at a time of my samples. So you can see the GFP being added as it goes on. It's a little hard to see, but what we'll begin to see is that the protein, the GFP, binds into the column. So after the first 500 microliters go through, we're going to add whatever is left in the centrifuge. Here I am adding the last bit of the samples, so you can see that the pellet is left and nothing else. It takes some time in order for these to go through. You can see in the video that the drops come at slightly different rates. Some come quickly, some take a little bit longer. So once you are clarified lysate, so clarified just means that you spun out all of the insoluble parts. So once your clarified lysate is pelleted and fully loaded onto the column, you're going to wash your column with wash buffer. The wash buffer has a little bit of imidazole and other buffers in order to keep your protein soluble. So what you'll see here is I'm going to add 500 microliters to each tube. And then you wait until all of the liquid, which is a little hard to see, is all the way to the bottom. So you can see that this is taking time. In the next segment, it's going to be much, much quicker. So this is the second wash. Make sure that you try not to disturb the bed. So if you look closely in this video, you can see that the sample bed or the light blue beads are not disturbed during this time. And so that's good. If you disturb them, you're likely to knock off some of your proteins. So each wash takes a minute or so. You want to make sure that you are paying attention to make sure that the beads don't dry out, but you also don't want to keep adding buffer over and over again. You want all of the buffer to drain to the bottom and then move on to the next wash. So you can see in the GFP, the center tube, that the buffer line is getting pretty close to the tubes. So now this one, I believe, is sped up. You can see that the continued washes, there are 6 total washes throughout this experiment. For reference, this took about 15 to 20 minutes. So now I'm going to use a chem wipe and dry off the bottom. You just kind of drag it along. Any excess moisture should come right off. So I'm adding a little yellow cap at the bottom so that when you add the elution buffer, which you'll see here in just a second, I'm going to add the elution buffer and pipette up and down, changing tips each time. So once you do that, you can see I'm going to let those sit for 5 minutes. I'm going to repeat that. So now you'll have a total of 300 microliters in there. So you want the cap on, otherwise it'll drip out the bottom. So you're going to pop it off and set it into a giant clear 2 ml tube. It would be good to have a rack on the side. So when you do that, some of the protein will start dripping off pretty quickly, the same as it has been. So once you have all 3 of those, you can see that the proteins are already starting to drip through, but we're going to come over and use the centrifuge. We're going to set it up and have balanced tubes in the machine. So then I'm going to put the lid on just to make sure nothing happens. I'm going to pulse it, and you can see for 4 or 5 seconds. You just want to kind of get it to spin up a little bit more. You do want to use this centrifuge as opposed to a quick spin. Once you do that, you can see that the beads are dry. And once the beads are dry, the purification is done.